Welcome to another deep dive into the latest longevity papers. We've got some really fascinating research hot off the press this week. Stuff from journals like Nature, Cell Reports, even some preprints from bioRxI.
Oh, very nice.
Yeah, and we're going to focus on what we think are the top 5 papers, the ones that'll really, kind of get you thinking.
All right.
We're looking at DNA methylation, mitochondrial dynamics, even the surprising role of cinnamon, and a new link between gut health and oxidative stress.
Wow, that sounds like quite the mix.
It is. So ready to see what the cutting edge of longevity research has to offer?
Absolutely. Let's dive in.
Okay, so let's jump right in with this paper that really kind of grabbed our attention. It's called Microglia Aging in the Hippocampus, Advances Through Intermediate States That Drive Activation and Cognitive Decline.
Okay.
And this one's a preprint. It came out just last week, January 3rd, on bioRxI.
Very recent.
Yeah, from researchers at UCSF. And it really takes a deep dive into how microglia, those brain's immune cells, change as we age.
You know, we often hear about brain cells just dying off as we get older.
Right.
But the study shows that it might not be that simple. You know, microglia, they don't just go from like young and helpful to like old and dysfunctional, right?
Right.
They actually go through these intermediate states.
Mm-hmm.
And it's these in-between stages that seem to be driving inflammation and cognitive decline, at least in mice.
That's a big deal. And
Yeah.
they actually, to confirm this, they use this really cool technique called heterochronic parabiosis,
Yeah.
which is essentially linking the circulatory systems of young and old mice.
Wow.
And they found that these intermediate microglia states also popped up in the younger mice when they were exposed to the older mouse's blood.
Fascinating. So it's like the younger mice are getting influenced by the older mice's aging process.
Exactly, yeah.
And that's where I think it gets really interesting for us, you know, thinking about implications. What if we could target these specific intermediate states and almost reprogram microglia to stay in a more youthful state, right? We could potentially slow down cognitive decline significantly.
That's an incredible thought. And now kind of shifting gears a little bit to another area that's getting a lot of attention in longevity, the gut-brain connection.
Yeah.
There's a paper in the journal Oxidative Stress that came out just yesterday, January 8th, and it's titled Low Input Redoxomics Facilitates Global Identification of Metabolic Regulators of Oxidative Stress in the Gut.
Okay.
And researchers from Sichuan University have developed a more accessible way to study something called the redoxome.
The redoxome, that's a mouthful.
It is, yeah.
But essentially, it's the landscape of oxidative stress in a cell.
Right, yeah, all those free radicals everyone's talking about.
Exactly.
So how does this new method help us understand the aging gut?
Well, they were able to study 5 different cysteine states with much less effort than traditional methods. And this is huge for the field because it just makes this type of research much more accessible, right?
Right.
And they used this method to look at the aging gut in monkeys.
Okay.
And they found that specific oxidative modifications, particularly SOH and SSG, were mainly affecting proteins involved in cell adhesion.
So why is cell adhesion so important in the gut?
Well, you know, it's all about maintaining a healthy gut barrier.
Right.
You know, think of it as like a wall that keeps harmful substances from leaking into your bloodstream.
Right.
And if that barrier breaks down, well, you get inflammation, you know, that can spread throughout the body and really accelerate aging.
So it's about more than just digestion. It's about protecting the entire system from that kind of chronic inflammation.
Exactly, precisely. And they also made a fascinating connection between fumarate,
Okay.
which is a metabolite involved in energy production,
Right.
and its ability to actually alleviate gut oxidative stress.
Mm-hmm.
And they even saw this in a model of colitis, which is, you know, chronic inflammatory bowel disease.
Wow.
And here's something else that I thought was really interesting. They found that calorie restriction actually reshaped the gut's redoxome
Interesting.
in a beneficial way.
Well, that's not surprising considering the amount of research that supports the benefits of calorie restriction. But this, gives us more insight into how it works at a molecular level.
Absolutely, yeah. Now let's move on to a paper that I think will spark a lot of discussion.
Okay.
It's from Aging Cell, published January 6th, and it's called Dietary Cinnamon Promotes Longevity and Extends Health Span via MTORC1 and Autophagy Signaling.
Cinnamon.
I know.
Really?
Researchers at the Washong University of Science and Technology found that cinnamon, might be a longevity booster.
That's fascinating.
Yeah, they found that cinnamaldehyde, which is the active ingredient in cinnamon oil, increased lifespan and boosted stress resistance in C. elegans, even in a model of Alzheimer's disease.
So we're talking tiny worms here.
Yeah.
But, I mean, how does this relate to humans and aging?
Well, what's really interesting is they linked these benefits to the MTORC1 and autophagy pathways,
Yeah.
two key players in the aging process.
Right, those pathways are always popping up in longevity research.
Exactly. And here's the kicker, right? Cinnamon seems to mimic the effects of dietary restriction, but without actually reducing food intake.
Now, that would be a game changer if it translated to humans. Imagine getting the benefits of calorie restriction without having to, restrict calories.
Right, exactly. Of course, we need more research to see how this applies to humans,
Right.
but it's definitely it's a very promising avenue to explore.
It's something to keep an eye on. All right, now let's move on to a paper that delves into a question that has puzzled researchers for a while.
Hmm.
Why do some men live exceptionally long lives with, better health than their peers?
Right.
And this paper was published on January 8th in Cell Reports. It's titled Methylome Analysis in Long-Lived Men Deciphers DNA Methylation Modifications Associated with Male Longevity in Humans.
Okay.
Researchers from the Cumming Institute of Zoology took a really close look at the methylation patterns of long-lived men.
So DNA methylation, it's like a set of switches that can turn genes on or off, right?
Right. Yeah.
And it plays a really crucial role in aging. You might expect that long-lived men would have a slower methylation aging rate, right?
Yeah.
Meaning their epigenetic clocks would be ticking more slowly. But they found the opposite.
Oh, that's interesting.
Long-lived men actually had a faster rate than long-lived women.
That's unexpected. So what's the explanation for that?
Well, the key is that they also found thousands of unique methylation changes that were specific to these long-lived men,
Okay.
independent of age and sex. And these changes, especially in genes like PIWL1 and EECT1, seem to be linked to disease suppression.
So it's not just about the speed of the clock. It's about, how it's programmed.
Exactly. Yeah.
That's fascinating.
This suggests that these men have a unique set of epigenetic hacks
Right.
that protect them from age-related illnesses.
So could identifying these patterns early on lead to personalized strategies for promoting healthy aging in men?
Absolutely, yeah. This study highlights the potential of understanding the epigenome to provide valuable insights into the aging process and perhaps even interventions down the line.
That's really exciting. Now let's bring it back down to earth a little bit with a paper that explores a dietary intervention that could help us combat one of the biggest challenges of aging: cardiovascular decline.
Oh.
And this paper was published in NPJ Aging on January 9th. It's titled Long-Term Intake of Tomogitake Mushroom, that's Pleurotus cornucopiae,
Yeah.
mitigates age-related cardiovascular dysfunction and extends healthy life expectancy.
Wow.
Researchers from Kumamoto University focused on the effects of these mushrooms, which are packed with the antioxidant ergothioneine.
Okay.
And they found some pretty impressive results in mice, right?
Yeah, they did. The mushrooms helped the mice maintain heart and blood vessel function as they aged. They even improved exercise tolerance, which is a huge indicator of overall health span.
Wow.
So, yeah, pretty impressive.
So how did these mushrooms achieve these results?
Well, the key mechanisms seem to be boosting NRF2 and HO1, reducing those pesky reactive oxygen species, or ROS, and dampening inflammation,
Yeah.
all things we want to see as we age.
Right, exactly. So should we all be adding tomogitake mushrooms to our diets?
Well, hold on a second. While the results are promising, we still need more research to determine the optimal dosage for humans. But this study just adds to the growing body of evidence that what we eat can profoundly impact how we age.
It really is incredible. This has been a really incredible deep dive so far, from brain cells and gut health to cinnamon and mushrooms. We're seeing that aging is a complex puzzle with many pieces.
It really is. And while we may not have all the answers yet, the research is advancing at an incredible pace.
It is.
Each of these papers contributes a valuable piece to the longevity puzzle.
And we have even more fascinating research to discuss, so stay tuned for part 2 of our deep dive.
Welcome back to our deep dive into this week's most exciting longevity papers. We've got a lot more ground to cover.
Oh, I'm ready for more. What's next on our list?
Let's talk about a paper that delves into the crucial relationship between aging and DNA repair. It's called Endothelial ERS-EC1 DNA Repair Deficiency Provokes Blood-Brain Barrier Dysfunction. And it was published on January 4th in Cell Death and Disease.
Okay.
Researchers at Amsterdam UMC investigated the role of ERCC1, a key DNA repair protein, in brain endothelial cells.
Okay.
Now, these are the cells that form the blood-brain barrier, which is essential for protecting the brain from, harmful substances.
Right. So if there's DNA damage in those cells, I mean, could that spell trouble for the brain?
That's exactly what they found. When ERCC1 was deficient in these cells, they saw an increase in something called senescence-associated secretory phenotype expression, which is basically a cocktail of inflammatory molecules that senescent cells release.
Wow.
And they also observed reduced blood-brain barrier integrity, meaning it was easier for harmful substances to leak into the brain.
So like a compromised blood-brain barrier could lead to accelerated brain aging?
That's right. And they saw this in their mouse model as well. Mice lacking ERCC1 in their endothelial cells showed signs of premature brain aging.
Right.
So this study provides a clear link between DNA repair and brain health.
Yeah, it makes you wonder if boosting DNA repair could be a way to protect the brain as we age.
It's definitely a promising area of research. Now let's shift our focus to a paper that examines a potential way to mitigate tau pathology,
Okay.
a hallmark of Alzheimer's disease.
That's a big one. There's so much research going on in that area.
There is. This paper was published in Alzheimer's and Dementia on January 2nd, and it's titled The Etiology and Prevention of Early-Stage Tau Pathology in Higher Cortical Circuits, Insights from Aging Rhesus Macaques. Researchers at Yale Medical School used aging macaques as a model, which is particularly relevant because their brains are more similar to human brains than, those of mice.
Right, so what did they learn from these primate cousins of ours?
Well, they found that age-related inflammation leads to calcium dysregulation in the brain, and this seems to be a driving force behind tau hyperphosphorylation.
Okay, so break that down for me a little bit.
Basically, the inflammation throws off the balance of calcium, which then causes the tau protein to become sticky and clumped together, leading to neuronal damage.
Oh, I see.
They even observed this seeding of tau pathology where it spreads across different areas of the brain.
Wow, so if we could reduce inflammation, could we potentially prevent or slow down that tau buildup?
That's the hope. And here's the exciting part. They found that by restoring calcium regulation in the brains of these aged macaques, they were actually able to reduce levels of a specific form of tau, p-tau 217, which is a key biomarker for Alzheimer's disease.
How did they restore calcium regulation? Was it some kind of invasive procedure?
Actually, they used pharmacological interventions, in other words, drugs.
Oh, interesting.
This suggests that there might be a way to develop targeted therapies that could protect the brain from tau pathology without resorting to invasive procedures.
That's a huge step forward in the fight against Alzheimer's. Now let's move from the brain to the heart. We have a paper that explores the role of a specific protein in maintaining cardiovascular health as we age. It's titled, TERT Prevents Obesity-Induced Metabolic Disorders by Promoting Adipose Stem Cell Expansion and Differentiation. And it was published on January 3rd in the journal Physiology.
Okay.
Researchers at the Marseille Cancer Research Center used a mouse model to investigate the role of TERT,
Okay.
the protein responsible for maintaining telomeres, those protective caps on the ends of our chromosomes.
Right. TERT, well known for its connection to aging. But this study focused on its role in obesity and metabolic health.
Exactly. They found that by increasing TERT expression in these mice, they could actually reduce metabolic disorders associated with obesity, even when the mice were fed a high-fat diet.
That's remarkable. So how did TERT achieve this?
It seems to be linked to a decrease in senescent cells in adipose tissue, which is where we store fat.
Okay.
Basically, TERT helped keep the fat cells healthier and functioning better, even under the stress of a high-fat diet.
So it's like TERT is playing a protective role against the negative consequences of obesity.
Yes. And here's where it gets even more interesting. They found that even when they inactivated the catalytic activity of TERT, meaning it couldn't do its usual job of lengthening telomeres, it still had some protective effects.
So it's like TERT has multiple functions. It's not just about telomere length.
Right. It appears to have other mechanisms at play. It seems to promote the formation of new fat cells, which helps to prevent the development of those unhealthy hypertrophic fat cells that are linked to metabolic dysfunction.
Fascinating. It seems like we're constantly discovering new things about these key players in aging.
Absolutely. Now let's dive into a paper that sheds light on the complex world of mitochondria, the energy-producing powerhouses of our cells.
Oh, mitochondria are always a hot topic in longevity research, right?
They are, yeah. They play such a crucial role in cellular health and aging.
They do. Uh, this paper, a preprint from Bioarxia, is titled "Miso Regulates Mitochondria Dynamics and mtDNA Homeostasis by Establishing Intermembrane Subdomains," and it came out on January 2nd.
Okay, break that down for me. What is MISO and why is it important?
MISO stands for Mitochondrial Intermembrane Subdomain Organizer, and it's a newly identified protein that seems to be a key player in regulating mitochondrial dynamics.
What do you mean by mitochondrial dynamics?
It refers to the way mitochondria fuse together and divide, which is essential for their health and function.
Right.
MISO orchestrates the formation of specialized subdomains within the inner mitochondrial membrane, and these subdomains are crucial for maintaining the balance between mitochondrial fission and fusion.
So if MISO isn't functioning properly, could that lead to mitochondrial dysfunction and accelerated aging?
That's what the research suggests. They found that by manipulating MISO levels, they could influence mitochondrial dynamics and even affect the stability of mitochondrial DNA, which is essential for their proper function.
This is cutting edge stuff.
Yeah.
It's incredible to see how scientists are uncovering these intricate details of mitochondrial biology.
It is.
It's like peeling back the layers of an onion, revealing more and more complexity with each new discovery.
I agree. And this deeper understanding is essential for developing interventions that can target mitochondrial dysfunction, which is a major hallmark of aging.
Well, this deep dive has been an absolute whirlwind so far.
It has.
We've explored everything from brain inflammation and DNA repair to the mysteries of telomeres and mitochondria.
And we're not done yet. We have even more groundbreaking research to discuss in the final part of our deep dive, so stay tuned.
Welcome back to the final part of our deep dive into this week's longevity papers. We've covered a lot of ground so far, from the brain and the gut to the heart, and those powerhouses of our cells, the mitochondria. But we're not done yet. There are still some fascinating studies we need to discuss.
I'm eager to hear what else is on the docket.
Let's start with a paper that takes a closer look at the connection between aging and the immune system, specifically a process called inflammaging.
Ah, yes, inflammaging.
Yeah.
That chronic, low-grade inflammation that seems to just increase as we age, contributing to all sorts of age-related issues.
Exactly. And this paper was published on January 3rd in the journal Inflammopharmacology. It's titled Stimulation, Regulation, and Inflammaging Interventions of Natural Compounds on Nuclear Factor, kappa B, NFKB pathway. A comprehensive review.
Okay.
Researchers from Barathear University dove into the role of the NFKB pathway, a key regulator of inflammation, in the context of aging.
So they're looking at how natural compounds might be able to modulate this pathway and potentially, put the brakes on inflammaging.
That's the idea. The review highlights a whole range of phytochemicals, those beneficial compounds found in plants, that have shown promise in preclinical studies for their ability to interact with the NFKB pathway and dampen down inflammation.
So it's like giving the immune system a much-needed timeout.
Exactly. And the exciting part is that many of these compounds are found in everyday foods.
Oh, like what? Give us some examples.
Well, there's curcumin from turmeric, resveratrol from grapes, and EGCG from green tea. Of course, we need more research to determine the optimal doses and long-term effects in humans.
Right.
But it is fascinating to consider that our diet could play a significant role in mitigating inflammaging.
It's amazing how powerful those everyday foods can be. Now let's move on to a paper that uses a unique model to study aging. It's titled Functional Analysis of Conserved C. elegans BHLH Family Members Uncovers Lifespan Control by a Peptidergic Hub Neuron. It was published in PLS Biology on January 6th.
Ah, C. elegans, those tiny transparent worms that have taught us so much about aging.
They really have. This study focused on a family of proteins called BHLH transcription factors, which are involved in controlling gene expression. And the researchers from Columbia University found that one of these factors, HLH15, was playing a role in lifespan regulation.
So by manipulating the activity of this one protein, they could influence lifespan.
That's right. They found that HLH15 was specifically controlling the activity of a type of neuron called AVK, which acts as a central hub for releasing neuropeptides.
Okay.
These neuropeptides are like signaling molecules that communicate between different cells in the body.
So this one neuron is essentially a control center for aging signals.
It seems that way. And here's the really interesting part. They found that when they knocked out HLH15, it actually extended the lifespan of the worms.
Wow, so by silencing those signals from the AVK neuron, they were able to slow down the aging process.
Exactly. It suggests that the AVK neuron might normally be sending signals that accelerate aging.
That's incredible. Of course, this is in worms, but it's still a remarkable finding. It could provide valuable clues for understanding how similar mechanisms might operate in more complex organisms like us.
Now, let's shift our attention to a paper that explores the fascinating world of extracellular vesicles, those tiny packages that cells release to communicate with each other.
Extracellular vesicles, so they're like tiny messengers traveling throughout the body.
That's a great way to put it. This paper was published in the journal Biogerontology on January 3rd. It's titled, "The Protein Cargo of Extracellular Vesicles Correlates with the Epigenetic Aging Clock of Exercise-Sensitive DNA Amphitage."
Mhm.
Researchers from the Hungarian University of Sports Science investigated the link between these vesicles and a specific type of epigenetic aging clock called DNA amphitage.
DNA amphitage, that's a way to measure biological age based on patterns of DNA methylation, right?
You got it.
And it's particularly sensitive to the effects of exercise.
Yeah, what's interesting is that they found a correlation between the protein cargo of these extracellular vesicles and the acceleration of this epigenetic clock.
So the contents of these vesicles reflect how quickly a person is aging, at least according to this particular clock.
Exactly. And they found that individuals with higher levels of aerobic fitness had different protein profiles in their vesicles, suggesting that exercise might be influencing the content of these messages that cells are sending out.
This is adding another layer to our understanding of how exercise benefits us at a molecular level.
Now, for our final paper, let's focus on a specific region of the brain that's known to be important for learning and memory and unfortunately also very vulnerable to aging, the dentate gyrus.
The dentate gyrus, that's part of the hippocampus, the brain's memory center.
Precisely. This paper was published in Nature Neuroscience on January 7th. It's titled, "Multimodal Transcriptomics Reveal Neurogenic Aging Trajectories and Age-Related Regional Inflammation in the Dentate Gyrus."
Okay.
Researchers from the University of Zurich used cutting-edge techniques to map the molecular changes that occur in the dentate gyrus as we age.
And what did they find?
They focused on neural stem cells, those special cells that can give rise to new neurons, and how they change with age. Interestingly, they found that neurogenesis, the process of creating new neurons, actually continues throughout life in the dentate gyrus.
So we don't completely lose the ability to make new brain cells as we age.
That's right. But it does decline as we get older. And they also found that age-related inflammation plays a role in this decline.
So inflammation is essentially putting the brakes on neurogenesis, contributing to cognitive decline.
Unfortunately, yes. But understanding these mechanisms gives us potential targets for interventions. What if we could find ways to reduce inflammation and boost neurogenesis in the aging brain?
That would be a game changer for maintaining cognitive function as we age.
I agree. This has been an incredibly stimulating deep dive. We've explored so many exciting avenues of research, from the intricate workings of our cells to the potential of natural compounds and lifestyle interventions to impact the aging process.
It's clear that the field of longevity research is rapidly advancing, and there's so much to be hopeful about.
I completely agree. It's an exciting time to be involved in this field, and I can't wait to see what breakthroughs the future holds.
And to all our listeners out there, keep exploring, stay curious, and let's continue to push the boundaries of healthy aging together. Until next time, keep diving deep.